I'm Dr. Linda Steingold. I am the Director of Dermatology Clinical Research at Henry Ford Health System and also a division head there.
So
So a lot has happened in acne, and this is probably the most common skin condition that we treat in our offices.
And we
know that prior to coming in, and even when they're already seeing us, a lot of our patients go to the internet to get their information.
There's a
There's a
lot
lot
of
of information on the internet,
but
is it all good information?
Well,
Well,
I guess we'll just
let the information speak for itself.
So is
it true
that if you've seen
one retinoid, you've seen them all? Honestly, do we really need a revolution in topical retinoids?
Well, the truth
of the matter is yes.
We have
new developments in topical retinoids that really are revolutionary.
First of all,
when we look at topical tretinoin, which was our first topical retinoid, we have a better formulation in a lotion that actually provides better tolerability than any of the other tretinoin formulations.
We have
tazarotene in a lotion formulation. Notice the percent, 0.045%. I want you to remember that tazarotene in the cream and gel and 0.05% is not FDA approved for acne, only for psoriasis.
But now
we have a lower concentration lotion
that
actually has an enhanced vehicle.
And with this
particular formulation, we actually get the same efficacy as the 0.1% cream, but we have a much better tolerability profile.
We also have
trifarotene, and this is a newcomer to the bunch.
This is specific for the
RAR gamma receptor, which is the predominant retinoid receptor in the skin.
In
clinical trials, it was studied on the face as well as the trunk, and it had proven efficacy as well as tolerability in all areas.
So is it
a myth that we can't use tretinoin and benzoyl peroxide together?
Well, let's look
at the data. Remember that adapalene is stable in the presence of both light and benzoyl peroxide, but that's not true of tretinoin. Tretinoin is actually inactivated by light, and it's inactivated by benzoyl peroxide. So if you have a patient that washes their face with a benzoyl peroxide cleanser, puts on generic tretinoin that was cheap and easy to get
by prescription, and then walks
outside, we have a double hit
because it's
inactivated by the benzoyl peroxide as well as the light.
We actually
now are able to mix these unmixables.
We have a
microencapsulated tretinoin at full strength, 0.1%, as well as a 3% benzoyl peroxide.
These
two active ingredients are actually microencapsulated in a silica shell.
What that
means is the active ingredients are actually encapsulated
with silica,
and there's multiple layers that actually allows for very slow delivery of each of the actives. By doing this, we can combine these two ingredients in the same tube, in the same vehicle, and have full activity of both of the actives.
This
was studied in patients with moderate to severe acne, all the way down to age 9. It's used once a day as compared to vehicle over the course of 12 weeks. When we look at the patients who came into this study, it's important to do this because we want to make sure that these patients really represent a typical acne group, and they do.
The average age
was around 20. It was a little more female than male, actually. We had a good distribution by race. Notice that about 90% of the patients had moderate disease, about 10% had severe disease, and they had about 70 lesions total on their face.
And what we
found was this fixed combination in a microencapsulated silica shell actually had up to 38% efficacy. That means getting patients to clear or almost clear. And by definition, it was a 2-grade improvement at least because they all started with moderate or severe disease. We had statistically significant efficacy in both of the phase 3 clinical trials.
Here's some
examples of patients who are in the studies. We see at baseline some pretty significant inflammatory diseases with a nice improvement by week 12. Here's another patient, especially on the temples. We see a nice improvement.
This patient
had really significant inflammatory disease. And looking at this patient, it might not be somebody that you would think about using a topical agent, but actually a nice improvement, especially in the inflammation with just monotherapy with a fixed combination.
When we look at the
tolerability actually was fairly well tolerated. These graphs can be difficult to read overall, but what we found is it's not much different than what we would expect from a well-tolerated topical retinoid.
So
So
we
we
have
have
a
a
triple
triple
combination. We
combination.
have a
We have
triple
a triple
combination that has
combination that has
undergone
undergone
phase
phase
3
3
clinical
clinical
trials.
trials.
It's
It's
clindamycin,
clindamycin,
1.2%, benzoyl
1.2%, benzoyl
peroxide,
peroxide,
3.1%,
3.1%,
adapalene,
adapalene,
0.15%.
0.15%.
Why should
Why
I
should
care?
I care?
Well, do
Well,
fixed
do fixed
combinations really
combinations
give us
really give
any
us any
significant
significant
benefits?
benefits?
And the
And
answer
the
is it
answer is
really
it really
depends on the
depends
fixed
on the fixed
combination
combination
because
because
they're
they're
not
not
all
all
created
created
equally.
equally.
And I want to
And
take
I
you
want to
back and
take you
give you a
back
little
and
bit
give
of
you
a
a little bit of
history
a
lesson
history lesson
on what we can
on what we
expect
can
from
expect
fixed
from fixed
combinations.
combinations.
The
The
bar
bar
used to be
used to be
really, really
really
low to
low
get a
to
fixed
get a fixed
combination FDA
combination
approved.
FDA
For
approved. For
instance, when we look
instance,
at
when
the
we look at
data
the
from the
data
fixed
from the fixed
combination of
combination of
clindamycin and benzoyl
clindamycin and benzoyl
peroxide,
peroxide,
clindamycin 1%,
clindamycin
benzoyl
1%,
peroxide
benzoyl peroxide
5%,
5%,
BID gel,
BID gel,
what's
what's
interesting is
interesting is
it was
it
not
was not
statistically
statistically
superior to
superior
benzoyl
to benzoyl
peroxide
peroxide
for the
for the
comedones,
comedones,
only for the
only for the
inflammatory
inflammatory
lesions.
lesions.
Interesting.
Interesting.
What about the
What
other
about
fixed
the other fixed
combination?
combination?
Clindamycin,
Clindamycin,
1% benzoyl
1% benzoyl
peroxide, 5%
peroxide,
in the
5%
once
in the
daily
once daily
application.
application.
Well, guess what?
Well,
They did
guess what? They did
5 clinical
5 clinical
studies
studies
looking at this.
looking at this.
Only
Only
3 out of the
3 out of the
5 were
5 were
statistically
statistically
significant
significant
for the fixed
for the fixed
combination as
combination
compared
as
to just the
compared to
benzoyl
just the benzoyl
peroxide.
peroxide.
Interesting.
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 What about the
What about the
clindamycin
clindamycin
tretinoin fixed
fixed
combination?
combination?
Well, actually,
Well,
when we look
actually,
at the
when we
fixed
look at the fixed
combination,
combination,
when we look
when we
at
look at
inflammatory
inflammatory
lesions,
lesions,
it
it
was
was
not
not
statistically
statistically
superior to
superior to
clindamycin
clindamycin
alone.
alone.
And when we look at
And when we look at
comedones,
comedones, it was not statistically significantly
better
better
than the
than the
tretinoin
tretinoin alone.
The bar
The
was not
bar
that
was not
high.
that
Do you
high.
guys
Do you
remember that?
guys
Isn't that
remember
kind of
that? Isn't that kind
crazy?
of crazy?
Wow.
Wow.
We did
We
start
did
to
start
see
to
an
see an
improvement in the fixed
improvement in the fixed
combinations when we look at the
combinations when we look at the
adapalene
adapalene
0.1% and the
0.1% and the
2.5% benzoyl
2.5% benzoyl
peroxide.
peroxide.
Now we see a
Now we
fixed
see a fixed
combination that
combination that
was
really was
statistically
statistically
superior to the
superior to
individual
the individual
ingredients. And
ingredients. And we
we saw
saw
this
this
for
for
both
both
the
the
comedones
comedones
as well as the
inflammatory
lesions,
lesions,
and
and
we
we
saw
saw
it
it
for
for
the
the
overall
overall clearance
or improvement
or improvement
investigators global
investigators
assessment.
global assessment.
So that
So
brings
that
me to
brings
the
me to the triple
Why should
combination.
I
Why
care?
should I care?
Well, how does
Well,
this
how does
drug
this
do?
drug do?
Well, this
Well,
drug
this
in phase
drug
2
in phase
studies,
2
this was a
studies, this was a
fascinating
fascinating
study because
study
to
because to
get
get
a
a triple
triple
combination
combination
approved,
you have to
you
look at
have to look
the
at the
duads. You
duads.
have to
You
look
have to
at,
look at,
first of all, just the
first of all,
vehicle
just the
by
vehicle by
itself.
itself.
But then you have
But
to
then you
look
have
at
to look
the
at
adapalene
the
and the
adapalene and
clinda,
the clinda,
the
the
adapalene
adapalene
and
and
the
the
benzoyl
benzoyl
peroxide,
peroxide,
the benzoyl
the
peroxide
benzoyl
and the
peroxide and
clinda.
the
So you have to
clinda.
look
So you
at
have to look
the
at the
duads and
duads
compare
and
that to the
compare that
triple.
to the triple.
And what we
And what we
found
found was,
very
very
interestingly,
interestingly,
each of the
each of the
individual
individual
ingredients
ingredients appear to contribute almost
to
equally
the
to
overall
the overall
efficacy of the fixed
efficacy of the fixed
combination. Each of them
combination. Each of them
contributes about
contributes about
15%.
15%.
And when we look at
And
the
when we look at the
overall
overall
efficacy of the
efficacy of the
triple,
triple,
we would
we
expect
would
if each one
expect if each one
contributed
contributed
15%, we might
15%, we might
expect 15,
expect
30,
15, 30,
45%.
45%.
We actually
Well,
saw
we actually saw
53%
53%
of patients
of
getting to
patients
clear,
getting to
almost
clear,
clear.
almost clear.
That's pretty
That's
good in
pretty
terms of
good
the
in terms of the
numbers.
numbers.
When we look at the
When we look at the
clinical
photographs, we
a
see
nice
a nice
improvement
improvement in the
inflammatory
inflammatory
lesions
lesions as well as the comedones.
So this
So
drug
this
was
drug
taken
was
into
taken
phase 3
into phase
clinical
3
trials
clinical
where we
trials
had
where we
two
had two
phase 3
phase 3
studies
studies
looking at the
looking
triple
at the triple
combination,
combination,
now
now
just
just
compared
compared
to
to
the
the vehicle
vehicle
itself.
itself. And what we
And what
found
we found
when
was,
we look at
when
the
we look
data,
at the data, compare phase 2
to the two
to the
phase
two phase
3s,
3s,
we now
we now have a topical agent that
over
gets
half
over
of the
half of the
patients all the way
patients
to
all the
clear,
way to
almost
clear,
clear,
almost clear,
especially when we're
especially
talking
when we're
about
talking
patients who
about
came
patients
in
who
with
came in
moderate
with
to severe
moderate to
disease.
severe
We
disease.
now have a
We now have a
topical
topical
drug
drug
that
that
has
has
higher
higher
efficacy
efficacy
than
than
any
any
other
other
topical
topical agent
or oral besides
besides
isotretinoin.
isotretinoin.
So we're
So
seeing
we're
these
seeing
patients
these
had
patients
about
had about
50% clear,
50%
almost
clear,
clear at the
almost
end of
clear at the
12
end of
weeks.
12 weeks.
Does it work
Does
for
it
both
work for
type of
both type of
lesions? Does it work
lesions?
for
Does
the
it work for the
inflammatory
inflammatory
lesions
lesions
and
and
the
the
comedones?
comedones?
Actually, it
Actually,
does.
it
And what we
does. And what
found
we
was we
found
had up
was
to
we had up to
80%
80%
improvement
improvement
in the
in the
inflammatory
inflammatory
lesions,
lesions,
which
which
means
means
the
the papules and the
pustules,
pustules,
and we had over
and we had over
70%
70%
improvement
improvement
in
in the
the comedones.
comedones.
So it works for
So
all
it works
type
for
of
all types
lesions.
of lesions.
But now we have
But now we have
drugs
three
together.
drugs
What about the
together. What about the
tolerability?
tolerability?
Actually, on
Actually,
average,
on
this drug
average,
was
this drug was fairly well tolerated, pretty much what you can expect from seeing the doubles.
And
And
the
the
mean
mean
tolerability
tolerability
scores
scores
were
were
mild
mild
or
or
better.
better.
So now let's
So
go
now
on to
let's go
another
on to
hot
another hot
topic.
topic.
Can we reduce
sebum
sebum
topically?
topically? Well, if we go to our
If we go to our social media
and we look
and we look online,
absolutely.
absolutely.
If you use the
If
right
you use the right
moisturizer
moisturizer or the right cleanser, you can decrease your sebum.
Look at all
Look
the
at all
blogs,
the
they'll tell you
blogs,
how
they'll
to
tell
do
you how to
it.
do it.
But the problem is it's just not true.
Well,
No matter
no matter how well you moisturize or how well you
not
cleanse,
going
you're
to
not going to
decrease
decrease your sebum production.
But what about
But what
topical
about topical
retinoids?
retinoids? I put those on, I get all dried out.
Doesn't that
Doesn't
decrease
that decrease my sebum?
No, it
No,
causes
it causes some local
dryness, but
it doesn't have an
know
effect that we know
on
of on sebum production.
So when we look
So
at
when we look at what we can do topically, we can normalize follicular hyperkeratinization. We can decrease inflammation. We can kill the cutibacterium acnes. But we haven't been able to decrease sebum topically. That's the fourth pillar in the pathogenesis of acne that we can't do topically.
We can do it
orally
with hormonal therapies, with
spironolactone,
oral
with oral
contraceptive
pills,
but we haven't been able to do it topically. We now have a topical agent that at least in vitro
has been shown to potentially inhibit sebum production.
And with that
inhibition of sebum production, we also find a downregulation of pro-inflammatory cytokines and overall an inhibition of inflammatory pathways.
The way this drug works is it directly competes with the androgen receptors
in the
sebaceous gland,
and it
competes with
dihydrotestosterone, so it blocks those receptors that dihydrotestosterone cannot block
and cause the
enhanced sebum production and enhanced inflammation.
When we look at
the structure of clascoterone, we find, well, it looks kind of similar to dihydrotestosterone, but that makes sense because it's competing with the same receptor,
and it looks
actually fairly similar to spironolactone.
So this drug was studied. It's a topical drug. It's a BID cream. It was studied in patients using it actually twice a day over the course of 3 months or 12 weeks.
And then those patients went into a long-term safety study.
What's nice in the
long-term study was they actually not only used the drug on the face, but they also used it on the trunk.
So how did it do?
Remember, we always do 2 phase 3 clinical trials. We want to make sure that the data is believable. We want to make sure it's reproducible.
And what we found
was
this drug
hit all its endpoints.
We saw up to about a
20% reduction
or
20%
improvement
in patients getting clear or almost clear with at least a 2-grade improvement. We also saw it was statistically significant in reducing not only the papules and the pustules, but also the comedones. And this was true in both of the clinical trials.
When we look at the
side effect, this drug actually was fairly well tolerated and not that much different from the vehicle. In fact, when you look at edema, erythema, pruritus, the vehicle was actually higher than the active drug.
We also have
an open label extension. We do these open label extensions mainly for safety.
And the reason
is we have these skin diseases that can last for decades.
And we study
something for 3 months.
But the question is,
what happens? Unfortunately, they didn't cure you after 3 months. What happens if we use these drugs for longer periods of time?
And what we found
was in the open label long-term extension study, where patients went on and off drug depending on whether or not they got to clear or almost clear. We didn't identify any new treatment-emergent adverse events that were not identified in the phase 3 trials.
Local skin
reactions. Actually, this drug seemed to be well tolerated, and it was well tolerated not only on the face, but it was also well tolerated on the trunk.
Here's an example
of a patient baseline
in week 12. And we can see a reduction of inflammatory lesions. In
reality, we study these drugs as monotherapy in clinical trials because we have to show that this drug
can hold its
own. It can have efficacy if used by itself. In real life, I would say most of these drugs are going to be used in combination, especially clascoterone.
So bottom
line is our understanding of acne continues to evolve.
Our
facts of today may become fictions of tomorrow. Unfortunately, we have a good pipeline of new drugs that we can expect in the future.
So thank you so much.
Thank you so much.